All right. Hello, everybody. Welcome to Science Daily Challenge, Day 1. So today we'll be going through the biology questions, specifically focusing on mitosis. So our first question states, after 2 rounds of cell division, there are 4 daughter cells. So this is actually going to be a true statement. And basically what happens is after 1 round of cell division, there's going to be 2 daughter cells. And now that you have 2 daughter cells from 1 round of cell division, right, we said in the question that there's going to be 2 rounds of division in total. So now each of those 2 daughter cells will undergo division and thus producing a total of 4 daughter cells. There's also a formula that's like 2 to the power of n equals the number of daughter cells, where n equals the number of rounds of cell division. So with 1 round of cell division, 2 to the power of 1 is 2. 3 rounds of cell division, 2 to the power of 3 is equal to 8. Question 2. Before a cell divides, it must replicate its DNA. This is going to be a true statement, so A should be correct. And the reason for this is because if a cell does not replicate its DNA, then the new cell that's being created will not be able to be functional, right? The cell needs its DNA. The DNA is going to code for proteins, and the proteins are essential for the life of the cell. And so if the cell does not replicate the DNA, then one daughter cell will not have that DNA, and thus it will not be functional. Number 3, during mitosis, the chromosomes are in a loose, uncoiled state. So this is going to be a false statement. And the reason for this is because during mitosis, the 46 chromosomes, they're moving all over the cells, right? They're moving, across the metaphase plate, and they're being tugged apart, etc. And if these chromosomes are in this loose, uncoiled state, and they're being pushed around and dragged, in that specific scenario, these chromosomes would be tangled up so easily, and there's a very high likelihood that they could be, torn or ripped apart. Therefore, during mitosis, the chromosomes are actually in a very condensed, supercoiled state. Whereas in the rest of the cell cycle, so specifically during interphase, when the cell is not going through mitosis or periods of cell division, the chromosomes are going to be in this more coiled, uncoiled and loose, relaxed state. Question 4, what is the first stage of mitosis? The first stage of mitosis is going to be prophase. So there's a helpful acronym that's basically IPMAT. And so the cell cycle is composed of interphase and then mitosis. So interphase is basically like, think of it like the normal cell activity when the cell is not dividing. And then PMAT for mitosis represents the 4 distinct stages: prophase, metaphase, anaphase, and then telophase. Lastly, question number 5, after mitosis, the 2 daughter cells are, and this should be A, that they're identical. Because these 2 daughter cells, the purpose of mitosis is to create two new daughter cells that are basically identical to each other and identical to the original parent cell. And so this is why mitosis is important for things like regrowth, or if cells get damaged, be able to regenerate those cells. This is in contrast with another process called meiosis, which we will explore in more greater detail. With meiosis, this is where you are producing, haploid cells, and each of these cells are different from each other. And these haploid or sex cells are important for genetic diversity. But again, with mitosis, so there's a T here, with mitosis, we want these 2 daughter cells to be very identical. So hopefully you all did very well on this first challenge, and I will see you all tomorrow. Thank you.